Bismillahirrahmanirrahim. This is Dr. Nighat Naeem from the Department of Biochemistry. Today I am delivering my second lecture about the cholesterol metabolism, and today's topic is regulation of cholesterol synthesis. This topic is very important because it is often asked in UHS exams. So let's move on. There are the 5 different types of metabolic controls which are helping in the regulation of cholesterol synthesis. The number 1 is sterol dependent regulation of gene expression. Second is enzyme degradation. Then is the dephosphorylation, the third factor. Fourth is the hormonal regulation, and fifth is the drug inhibition. One by one, we'll study all these 5 factors. The number 1, sterol dependent regulation of gene expression. Here you are seeing that the enzyme in the phosphorylated form, it is inactive, and it will be active when it is in dephosphorylated form. How the gene expression is controlled? This is controlled by a protein called SREBP. This is the sterol regulatory element binding protein. When the sterol level in the cell it is low, what happens? This SREBP, which is present on the endoplasmic reticulum, smooth endoplasmic reticulum, in association with another type of protein called SCAP. SREBP, SCAP. SCAP is cleavage activating protein. SREBP, cleavage activating protein. When the sterol level in the cell it is low, these two associated protein will move to the Golgi complex, and here in the Golgi complex there will be the action of proteolytic enzyme, and the SCAP will be detached from SREBP. Then SREBP will be converted into the solubilized form, and it will enter into the nucleus. In the nucleus, it will bind at the DNA at SRE, sterol regulatory element. Element here in the gene expression, we use the word element for a part of the DNA which is involved in the regulation of gene expression. So here SRE will be bounded by the SREBP, and there will be the transcription and the translation, and there will be the formation of HMG-CoA reductase. However, when the sterol level in the cell it is high, then this complex SREBP and SCAP will be bounded by another protein called INSEG. INSEG is insulin induced gene protein. The bounded form of this INSEG, SCAP and SREBP is not shown in this diagram. However, it is shown in the some new editions of the Lippincott. This picture actually I have taken from the 6th edition, but in the 7th edition you will see that the INSEG protein is also shown here. So come to the topic. Cholesterol, when it is high, what happens? The INSEG will bound here, and this bounded INSEG, what is its action? It will not allow this complex to move to the Golgi, right? And it will remain here, and the bound form of SREBP, SCAP and INSEG will not go to the DNA, where it will not bound to the SRE, and there will be the no transcription and no translation. So this is the sterol dependent regulation of gene expression, in which there is the main role of protein SREBP. The second factor which effect on the cholesterol metabolism is the enzyme degradation. Here again the insect protein will play a role in down regulation of HMG-CoA reductase. When the sterol levels are high, it will bind with the reductase, HMG-CoA reductase, and its binding will lead to the ubiquitination and proteasomal degradation. Now the third factor is the sterol independent phosphorylation and dephosphorylation. Here you are seeing that the HMG-CoA reductase, when it is dephosphorylated, it is active, and when it is phosphorylated, it is inactive. Its phosphorylation is done by a protein. This is the AMPK. AMPK is AMP activated protein kinases. AMP activated protein kinase. Kinase is the enzyme which will phosphorylate the protein. This phosphorylation AMPK is itself will be active when it is phosphorylated, and its phosphorylation again is done by a protein. This is its phosphorylation is done by AMPKK. AMPKK is AMP activated protein kinase, right? And its dephosphorylation is done by the phosphatases, protein phosphatases, which will dephosphorylate AMPK, and dephosphorylated form of AMPK will be inactive. So when the AMPK will be inactive, there will be the no phosphorylation of HMG-CoA reductase. How this HMG-CoA is dephosphorylated? It is done by the protein phosphatases. Phosphatases means the phosphate will be removed from the HMG-CoA reductase. And you are seeing here in the diagram that protein phosphatases is stimulated by a hormone insulin. And insulin, you know that this is the anabolic hormones, and it will be released from the pancreas when the body is in the well-fed state, and when the cells they have a lot of glucose, and the glucose will be metabolized to the acetyl-CoA. So the acetyl-CoA, as you know, that this is the substrate for the substrate for the cholesterol synthesis. So when there is the abundant acetyl-CoA, HMG-CoA reductase will be active. And also here you are seeing that there is the also the role of glucagon. When there is the glucagon means when the cells they are not in the well-fed state, they are in the fasting state, and there is then the CMP will be formed, and which will be negatively or down regulate the HMG-CoA reductase. It will inhibit protein phosphatases. then the fourth factor which influence the cholesterol synthesis, regulation of cholesterol synthesis, is hormonal regulation. Hormones, insulin, as you have studied in the last slide, increase in the insulin, and also there is another hormone, thyroxine. They both favor up the upregulation of expression of the gene. And glucagon and glucocorticoids, they have opposite effect, means they down regulate the expression of HMG-CoA reductase. Drug inhibition, the statin drugs, these statin drugs, they are the actually the comparative or structural analogs of HMG-CoA reductase. They bind irreversibly with the enzyme HMG-CoA reductase and will inhibit its activity. These drugs, they are used in the patients to lower the use in the patients of hypercholesterolemia to lower the plasma cholesterol level. So in this slide we will study what are the factors which affect the cholesterol balance at cellular level. First, we will study what are the factors which increase the cell cholesterol. Increase in the cell cholesterol is by the uptake of lipoprotein particles which contain cholesterol. And the second is uptake of free cholesterol from these lipoprotein particles which have been taken up by the cells. The third factor is cholesterol synthesis, and the fourth one is hydrolysis of cholesterol ester by the enzyme cholesterol ester hydrolases. All these four events, they increase the cell cholesterol. Now, what are the things or what are the events which decrease the cell cholesterol? The decrease in the cell cholesterol is by the efflux of cholesterol from the membrane to the lipoprotein particles called HDL, esterification of cholesterol by the enzyme ACAT, acyl-CoA cholesterol acyl transferase, and the utilization of cholesterol for the synthesis of steroid hormones. So this is the diagrammatic representation of the last two slides, in which I have explained you how the unesterified cholesterol pool is maintained in the cell, especially in the membrane. See here, the LDL particles, they are either taken up by the receptors or some non-regulated pathway. They entered into the cell, they are taken up by the lysosomes, and which may be converted into the endosomes, and from the lysosomes this cholesterol is deesterified and is then enter into the cholesterol pool. Another factor is the cholesterol synthesis, and another factor which increase the cholesterol level in the cell is the deesterification of the cholesterol ester by the enzyme cholesterol ester hydrolase. Now, what are the things which decrease the cholesterol level in the cell? This is the cholesterol uptake by the HDL particles. HDL particles by the via ABCA1, ABCG1 or SRB1. And also I have told you in the previous slide that the cholesterol may be taken up from the membrane by the by esterification, and the enzyme here is ACAT. So this was all about my today's lecture. Best wishes and good luck.